Hello dear students, I am Dr. Syed Irfan Raza and today we are going to discuss a very important cell organelle which is known as lysosome. In this lecture we will study the detailed definition of the lysosome, what are lysosomes, formation, how they are formed in the cell, containing enzymes which are present in the lysosomes, their functions, related diseases, and at the end we will discuss few of the MCQs which are related to the lysosomes. So let's start from the definition of the lysosomes. In fact, lysosomes is a membrane bound cell organelle that contain digestive enzymes named as hydrolases. So in this GIF we will see that how the hydrolases containing membrane bound organelle which is termed as lysosomes are manufactured, and the enzymes which are present in the lysosomes are synthesized basically from the endoplasmic reticulum. These enzymes are synthesized and then they are transported to the Golgi apparatus, and from the Golgi apparatus a vesicle like structure which is known as lysosomes are formed. And when the bacteria or viruses gets entry by the process of phagocytosis, phagosomes are formed. They move fuses with the phagosomes and digest all the bacteria and the viruses. So lysosomes are very important. And lysosomes, why they are important and how they digest all the bacteria and viruses and what are the other functions, we'll see in the next slide. But they are containing some specific type of hydrolases that are enzymes which can digest protein of the viruses and bacteria, which can digest nucleic acid of the bacteria, which can digest lipids, and which can digest carbohydrates or glycogen as well. So they are containing a group of different enzymes, group of enzyme hydrolases, and these hydrolases contain number one, a glucosidase enzyme which can digest glycogen or the carbohydrates. Number two enzyme hydrolase cathepsins which can digest or hydrolyze proteins, and then they contain lipases which can digest lipids, and then they contain ribonucleases which can digest nucleic acid or RNA specifically. That's why the lysosomes contain this many enzymes. Number one, glucosidase, alpha glucosidase, which acts upon glycogen and digest it. Then cathepsins, which digest proteins. Then lipases, which digest lipids. Ribonucleases, which digest RNA. So the lysosome contain enzymes which are termed as a hydrolases. What are the main functions of the lysosomes? To see the main functions of the lysosomes, they are involved in various cell processes, and we in this slide will cover only two of the main processes or the functions. Number one, they break down excess or worn out cell parts, that is to maintain the cellular compounds in a dynamic state. For example, if there is a mitochondria which is old, which is not functional, lysosome will come in action and will digest the old mitochondria, ribosomes, or whatever, the to maintain the dynamic state of the cell, that the new mitochondria shall be there, the old mitochondria should be worn out. So lysosomes play a very important role in maintaining the cellular compounds in a dynamic state. The second important function is that they may be used to destroy invading viruses and bacteria. So let's see what's happening here. So for example, if the virus bacteria are entering by the process of phagocytosis, phagosome is produced containing the viruses and bacteria. Lysosome will come in action and they will fuse to the phagosomes and will digest immediately all the viruses and bacteria. On the other hand, this lysosome can move to the old cell organelles like mitochondria and will digest the mitochondria as well. So lysosomes have two main important functions. Number one, maintaining the cellular compounds in a dynamic state. Number two, they may be used to destroy invading viruses and bacteria. That's great. So what is lipofuscin, the age pigment? Look at this young girl. In fact, it's not related to the young, or it might be some genetic disorders, this child belongs from Pakistan, and myself seen almost 3 to 4 patients the same family. There are some dark pigments you can see on the cheeks, some dark pigments, and when we had a biopsy, we observed some dot like structure, dark pigmented bodies, and these pigmented bodies are in fact residual bodies, certain residual products rich in lipids and proteins, collectively known as lipofuscin. So these rich bodies are basically lipofuscin, and they get accumulate in the cell as a result of wear and tear pigments, which has been implicated in aging process basically. But in some of the families having genetic disorders in the very young age, there can be some dot like structures or brown pigmented bodies can be appear on the skin, whether it's a face, whether it's a hand or leg, whatever. The skin manifests some of the dark pigmented bodies. You can see these marks. These are brown in colors. They can have some, when the biopsy was taken or FNAC was performed, we observed some of the tissues containing these dark pigmented bodies, which are known as lipofuscins, and they are related to aging process. So as the cell dies, the lysosome ruptures and release hydrolytic enzymes that result in postmortem autolysis as well. For example, if there is an injury, what will happen? Cell will burst or cell will rupture. Some of the lysosomes will come out, and lysosome will get rupture, and all the hydrolases will come out and in the mainstream, and they will affect the residual bodies, or they will affect the vicinity, all the tissues and other cells in the present in the vicinity of that wound, or in the post-mortem autolysis you will find some hydrolytic enzymes, they are working there, and they are affecting the proteins and the lipids and the carbohydrates and number of other products of the cells. So that's why lipofuscin is basically the dark pigmented bodies which are basically related to the aging process, and it is because of the accumulation of the wear and tear pigmented bodies. Here you can see in this histopathology diagram as well. So if the lysosomes, in fact that all these digesting enzymes of the cellular compounds are which are present in the hydrolases in the lysosomes, they are confined to the lysosomes in the best interest of the cell. Why? For example, if the lysosomes gets ruptured, what will happen? The enzymes will come out, and they will affect the important compounds, that is glycogen and carbohydrates and proteins in the cytosol of the cell, and they will damage all the protein, because enzymes will not see whether the proteins are of our own cell or carbohydrate glycogen of the cellular or non-cellular, whether there's antigen or itself. It will digest everything. That's why if the lysosomal enzyme escape from the lysosome, they will get entry into the cytosol, and where they will destroy the functional macromolecules of the cells, like proteins, like other carbohydrates, like other lipids. And if they are affecting like this, it will produce number of clinical manifestations, like release of lysosomal enzymes can produce arthritis. Release of lysosomal enzyme can result in muscle diseases, and release of some lysosomal enzyme can cause some allergic disorders. So lysosomal enzymes, if they get released to the cytosol, they may result in arthritis, muscle disease, or allergic disorders. They are the key clinical features. Look at this child. He is suffering from eye cell disease. What is eye cell disease? In fact, the child is from Pakistan. Observe this child as well. Inclusion cell disease. Eye cell disease means inclusion cell disease, also referred to as mucolipidosis 2 or ML2, is part of the lysosomal storage disease family, because it's basically a family lysosomal storage disease family. That is the number of diseases which are in this family. So eye cell disease is one of the disease of this family, and result from a defective phosphotransferases. Defective phosphotransferases. Transferases mean addition of phosphate group to any specific moiety like protein. So phosphotransferases, an enzyme of the Golgi apparatus. This enzyme transfers phosphate, that is phosphotransferases, that is phosphate, to mannose residue on specific proteins. So basically this enzyme is present in the Golgi apparatus, and the function of Golgi apparatus is that they transfer mannose to the some of the proteins. And if this enzyme is not present, some of the enzyme will not be able to have mannose on their surface. Resultantly, those enzymes will not be able to move to the lysosomes. So basically, mannose 6-phosphate serve as a marker for the proteins to be targeted to lysosomes within the cell. So let's explain this thing. For example, if this is the cell, look at this. This is lysosome, and these blue color marked. Can you see the blue? Can you see the blue here? The blue. These are the some of the very important enzymes. And normally these enzymes which are blue in color, small dot like structure, they have some red parts, that is mannose 6 phosphate here on the surface, and these mannose particles are added by the Golgi apparatuses, and they transfer these proteins here, and these proteins get entry to the lysosomes in this way. If these mannose particles are not present here, if these are not present here, for example, this protein is not present here, there is no mannose here, this protein will not enter into the lysosomes. Resultantly, this lysosome will not be able to digest specific type of proteins because the enzymes are not there, because these enzymes are not there. Why it's not there? Because there is no mannose. So this is basically because of the absence of the mannose 6 phosphate enzyme, which is basically present in the Golgi apparatus. So the resultant disease is eye cell disease. So mannose 6 phosphate serve as a marker for the protein to be targeted to lysosome within the cell. That's why we say that inclusion cell disease is a rare condition due to the absence of certain hydrolases and a defect in the protein targeting, because protein cannot enter to the lysosome as an enzyme cannot reach to the lysosome. Resultantly, a clinical manifestation can be seen, which is known as eye cell disease. So now we see a couple of MCQs. Number one MCQ is a male patient age 58 year with light brown pigmented marks on the skin which were rich in hydrolases. Those marks were rich in hydrolases. Skin specialist or dermatologist diagnosed as the aging marks, which cell organelle may be involved in the underlying clinical manifestation. Cell organelle is asked. Which cell organelle can be involved because of the brown pigmented marks appeared on the skin? Number one, Golgi complex. Number two, lysosomes, mitochondria, nucleus, or peroxisomes. Obviously, the answer is lysosome, because lysosomes are involved in the brown colored pigmented bodies on the skin. Number two question is, mannose 6 phosphate serves as a marker for protein to be targeted to. If the mannose 6 phosphate is not present, protein cannot enter to which cell organelle? Number one, Golgi complex. Number two, lysosomes. Number three, mitochondria. Number four, nucleus. Number five, peroxisomes. Which is the right answer? Yes, it's lysosome. Lysosome is basically involved. Then, as a result of cellular wear and tear, lipofuscins are observed due to non-functional what? Non-functional Golgi complex, lysosomes, mitochondria, nucleus, or peroxisomes. The answer is again lysosomes. The last MCQ, lysosomal cathepsins work on. Lysosome contains number of enzymes which can digest lipids, which can digest proteins, which can digest glycogen, which can digest nucleic acid or RNA. Cathepsin is one of the enzyme as a hydrolase is present in the lysosome. Cathepsins work on what? Proteins, they work on DNA, they work on RNA, lipids, or carbohydrates. Yes. Which is the right answer? The right answer is proteins. So cathepsins work on protein. Dear students, you should know all the enzymes which are present in the lysosomes, and they work on different moieties like protein, RNA, lipids, or carbohydrates. You should learn all the enzymes respectively. Thank you very much for with me here. Listen this lecture, and if you can subscribe my channel Biochemistry with Dr. Irfan Raza, and hit the bell icon button as well, so that you can get my latest videos. Thank you very much and take care.